Good morning to all. Today we're going to discuss about the slide history of biotechnology. The technology that uses living organisms to produce and improve a desired products. With the help of plants and animals, you get a lot of products. That products is known as biotechnology. Biotechnology is nowadays the application of scientific and engineering principles to the processing of materials by biological agents, and it provides a goods and services. The term biotechnology was coined by Carl Erke. So various innovations have several practical applications in environmental protection and protection of natural resources like forest and everything, earth and forest, and in welfare of domestic plants, animals, and also human beings. The next slides shows the history of biotechnology. The history of biotechnology shows the traditional biotechnology as well as modern biotechnology. The traditional biotechnology has been used for thousands of years to produce a food and also a health care products. Today, modern biotechnology enables to use to develop a improved products. So all the improved products are safely, and the biotechnology is one form of another has flourished a prehistoric times. During the 3000 BC to 3000 BC, old people used to make bread, fermentation of fruit juices, and plant extrudents or to produce various alcoholic beverages with the help of yeast. The next slide shows the evolution of pre 20th century of biotechnology. 1770, Lavoisier gave a basis for alcoholic fermentation. How the alcoholic fermentation process takes place with the help of chemical process. 1798, Edward Jenner, first viral vaccine to inoculate a child from smallpox. In 1838, protein discovered by Gerardus Johannes and Jones Jacob. In 1871, Ernest Copier Saylor discovered enzyme invertase. The yeast having enzyme two enzymes, invertase and zymase. This invertase enzyme, which is used for making artificial sweeteners. In 1876, Louis Pasteur identified various microorganisms involved in fermentation. The next slide shows the history of 20th century of biotechnology. In 1919, the time biotechnology was coined by Carl Erke. 1928, discovery of penicillin by Alexander Fleming. The sources of penicillin or Penicillium notatum is now it is called as a wonder drug. So this is a very good antibiotic. Then in 1953, discovery of double helix structure of DNA. So structure of DNA, double helix model, Watson and Crick. In 1972, the discovery of restriction enzymes. The restriction enzymes are endonuclease, then exonuclease and ligase for various genetic engineering tools discovered by Arbor and Smith and Nathans. In 1975, production of monoclonal antibodies by Kogler and Milstein. The next slide says, after that continued 20th century, in 1978, production of human insulin, the insulin from E. coli. In 1979, development of artificial gene. They are discovered by H.G. Corona. 1983, use of TA plasmids to genetically transformation plants. So genetically modified TA plasmids are very important for tumor-induced plasmids. The tumor-induced plasmids caused by Agrobacterium tumefaciens. These tumefaciens are very important for genetically modified plants or transgenic plants. In 1986, a development of polymerase chain reaction, that is PCR, otherwise called a thermocycler. This thermocycler are discovered by Cray and Mullis. Nowadays, the coronavirus detection by PCR method. Then during the 2000, first plant growth of Arabidis thaliana is sequenced DNA genome. The first isolated plant genome get from Arabidis thaliana. The next slides shows during the 21st century history of biotechnology, 2001 Human Genome Project creates by the genome sequence. In 2002, plant crop like plant genome introduced in the common paddy plant Oryza sativa. In 2010, Robert Edwards developed a in vitro fertilization method in animals. During the 2016, stem cells are injected to stroke patients during with the help of stem cell therapy. During the 2017, blood stems are grown in a lab. So this we detailed study about the traditional biotechnology. The traditional biotechnology is a kitchen technology. This technology followed by our ancestors. The technology, what are the uses a lot of bacteria and microbes. They are used a preparation of our daily products like curd, ghee, and cheese. In the preparation of foods like idli and dosa are traditionally method of biotechnology. The preparation of alcoholic beverages like beer and wine are prepared from traditionally. These, this type of kitchen technologies are nowadays very scientific validation properties. The next slide shows modern biotechnology. The modern biotechnology is very important nowadays. The ability to change the genetic material and get a new products. All the new products are with the requirement through a rDNA technology, that is recombinant DNA technology. So the recombinant DNA technology, we get a lot of various engineering, genetic engineering, as well as recombinant technology. We get a lot of new products. The next one, the modern biotechnology, the ownership of newly developed technology and its social impact. Today, biotechnology is a more benefit around the world in the field of pharmaceutical companies, breweries, and agro industries. Finally, the modern biotechnology are these methods of genetic modification by cell fusion technology and recombinant DNA technology. So the next slides shows the focus of biotechnology. The various biotechnology biotechnology are used for various biotechnology field. They are so major focus of biotechnology are fermentation, then biomass, enzymes, biofuels, microbial inoculants, RDN technology, and various process of engineering. Fermentation for production of acids, enzymes, alcohols, antibodies, chemicals, vitamins, and toxins. So from the fermentation method, we get these products. And the second one is biomass. So biomass is a bulk production of a single. We get a lot of cells from isolated from a single cell protein from a blue green algae like chlorella and alcohol, and they are used as a biofuel. So it is you get more energy from the biofuel. So another one is enzymes. Enzymes are biocatalysts, but these enzymes are used as a sensor, biosensors in processing industry. Biofuels are get from production of hydrogen, alcohol, and methane. So all these are biofuel. Then another last one is microfill inoculants, that is biofertilizer and nitrogen fixers. Biofertilizers, we get various biofertilizers, chemical manures, and nitrogen fixers. And another one slide shows the plant and animal cells are production of secondary metabolites like alkaloids, terpenoids, and everything, and also produce monoclonal antibodies. So, with the help of RNA technology, the production of chemicals, enzymes, vaccines, growth hormones, antibiotics, and interferons. The interferons are protein based protein based drug. So from this RA technology, a lot of interferons are produced. The engineering during the engineering method, genetic engineering method. So the process of genetic, the biotechnologies are very important tools for effluent treatment from discharge of various waste and waste materials. Then all the effluent materials are from factories, industries. All these are recycling, and also drainage water, that is the drainage water should be recycling methods. That is a commonly they are called environmental biotechnology. So in modern biotechnology, different types of valuable products are produced with the help of microbiology division, biochemistry, tissue culture, genetic engineering, molecular biology, and immunology. Okay, thank you.